Yeah, just what time, we'll set something up afterwards. But we'll go out and grab a nice little date. Talk at 7?
Yes.
Good. All right. I was going to come. Good.
Yeah.
Yeah. That's good.
It's supposed to be that way.
I was told that. So I'm standing there.
And you just have to remember to start the voice as soon as I've already started the string so people
Oh,
can
the introduction is good.
No, it doesn't
Okay,
really,
excellent.
but they'll see you. And if you want to, if you stand in front of them.
All right, well, I may as well do that. We're still learning how to do this.
Yeah, it's seven years old, so and we just got the new projector, so it's music sensitive to it now. But I also think we shouldn't give everyone as much as we should every encourage people to come.
It's really true. It's funny. There was discussion about that with just like a science, ecology meeting, revolution meeting. Like they should traveling. It's all
Yeah.
but if we walk all over the
Exactly, that's what you're worried about. Because people think they watch, but they don't necessarily.
Welcome, everybody. It must be Monday at the Arnold Arboretum, and I'm really pleased to be able to introduce our speaker today, Professor Anurag Agrawal from Cornell University, who flew in from Montana. And you may wonder why, but he's spending a year of sabbatical, and Montana is one of the places he will be. The other place will be slightly warmer. Anyway, we've known each other for some time. I think we were trying to reconstruct back to 2002 when Anurag was an assistant professor at Toronto, and I was up to give a talk and a dinner with Spencer Barrett and all kinds of wonderful things. But it's really a great pleasure to have you here today. Many of you will know that he was originally invited in our public programs because of an amazing book he's written on monarchs and milkweed. And this book came out this year and has been reviewed by so many people with incredibly positive reviews. The reviews I've read would make most people blush. And it's a terrific piece of work because it's not just about the science. It's a way of actually reaching out to the public to bring people in to understanding how natural history and science can help shape our view of the world. So this evening, for those of you who haven't had enough, and I won't, I'll be there tonight, from the science talk, our public programs at 7 o'clock will have his book talk, so to speak. Anyway, let me give you a little bit of brief background. Anurag got his PhD at the University of California, Davis in 1999. And then got a master's degree in conservation biology at the University of Pennsylvania in 1994. Working backwards, he got his bachelor's degree at the University of Pennsylvania in 1994. So I assume that was a BA-MA program. He began his faculty career at the University of Toronto, where we met, and then he joined Cornell University as the assistant professor of ecology and evolutionary biology in 2004. With a cross appointment into the Department of Entomology. He's been at Cornell ever since, and he is now the James Perkins Professor of Environmental Studies at Cornell University. He's had many honors. He had an NSF Early Career Award. He's received the David Starr Jordan Prize, and more recently the Robert H. MacArthur Award from the Ecological Society of America. His papers, if you look over his career, are beautiful papers. They are rich in natural history and understanding and take on the topic of coevolution in ways that have been highly creative. I'll just mention one of my favorite papers, though, may not be one that's recent. It's about advocating for the breeding of Domatia. Do you remember that one? You do. Anyway, very early in his career, he actually did something on plant anatomy in a very specific sense, which I love, which is he talked about the role of these little hairy spots, parts of leaves that can be homes for, basically for arthropods and insects that can protect plants from the bad guys. And actually just a knowledge of structure and of natural history and ecology can come out in very interesting ways. I think that was that published in Nature or Science or something? Nature, yes. Anyway, so there are many things that are surprising when you start to look over his career and his record, and they're all wonderful. So without further ado, Anurag, welcome to the Arnold Arboretum.
Thank you, Ned, for that wonderful introduction. Thanks all of you for being here. It's been a great day already, and looking forward to the rest of it. This talk is about trade-offs and the importance of trade-offs in evolutionary ecology. And I think all of you who think about organisms in some way, shape, or form have considered trade-offs. There's perhaps the most central conceptual issue in terms of thinking about how organisms adapt to their environment. No organism can do everything. We think we all recognize that. And they occur at the level of individuals and the allocation decisions that individuals make. They occur among the different genotypes that occur within a population, within a species. And trade-offs are also important at the community or wider level, the species level. And the trade-offs perhaps dictate which species enter and coexist in a community versus those that don't. Trade-offs are often conceptualized as this giant Y-tube, where limiting amounts of resources for any organism are shunted one way towards some phenotype or some other way. And of course, and as we go through the talk today, I hope I'll convince you that of course it's more complicated than that. It's not simply a Y-tube, but it's a useful conceptualization. The reason I decided to give this talk on trade-offs is I was reflecting on the work I've done myself on various systems over the last 20 years or so, and I realized, somewhat surprisingly, that trade-offs have really motivated a lot of what I've done, a lot of questions that I've asked, but they've been different, and the questions and approaches have been different when I've considered phenotypic plasticity at one level, how an individual organism or an individual genotype might differ in its expressions depending on the environmental stimuli it receives, versus the genetic correlations we see within a population versus the species strategies we see. And it sort of dawned on me that often, especially as ecologists or evolutionary ecologists, we grab the conceptual framework or theory from one of those levels of organization and apply it or test it at some other level of organization where it may not necessarily be appropriate. So what I hope to do today is convince you of not only the general importance of trade-offs in biology, I think all of you are on that same page, but also hope I can provide a conceptual framework for thinking about trade-offs at these different levels of biological organization. Before we get there, I guess we should dive in, make sure we're all on the same page with a little bit of natural history. This is two individuals, the same genetic clone of a water fleid, organism in the genus Daphnia. And same genetic individual, they're genetically identical, they are clonal as are some plants. This one grew up in an environment lacking the chemical cues of its fish predators. This individual grew up in an environment with the chemical cues of its fish predator. Dramatic phenotypic difference. And the trade-off that you might imagine that occurs here is that this individual has expended energy that it could have expended perhaps on egg production in generating this head and tail spines, which are defensive. In addition, it's going to alter the swimming speed and perhaps other ecological interactions because this individual has that baggage, whereas its clone mate, which didn't grow up in the environment of those fish predators, doesn't have those protrusions. So at the level of phenotypic plasticity, an individual can generate different phenotypes, and there may be trade-offs associated with generating those plastically. If we think about classic examples of genetic trade-offs among different genotypes within a species, for a person like me, the macroptery versus microptery in insects comes up as a very common and classic form of a polymorphism, a genetically determined polymorphism that has associated trade-offs. In this case, we have genetically related individuals. I believe they're full sibs, so they're closely related, but through genetic determination, this individual has relatively small wings and highly reduced wing musculature inside the body, whereas this individual has highly developed. A dispersal morph versus a non-flying micropterous or non-dispersal morph. And the trade-offs associated here they're physical sometimes. They're pretty remarkable. If you cut open, I've actually never done this, but I've seen the pictures. These hoppers, there's actually physical space limitation inside of their bodies, where on this side, not only are there these large wings here, but the muscles associated with those wings take up a lot of space in that body, whereas that space is occupied by ovaries in this body here. So there's physical limitation, and certainly there's an energetic trade-off between allocation to those wings and wing muscles versus ovaries in the case here. Again, determined at the level of different genotypes within a species. If we move up one additional level of biological organization and take a lesson from life history theory, and this is something that spans plants and animals, there's perhaps a almost universal trade-off between propagule size and number. I'm showing you here images of each seed. Here is a different Asclepias, a different milkweed seed. And again, this occurs both within genera or across taxa, a general trade-off between the number of propagules, in the case of plant seeds that are produced, versus the size that they're produced. Those that tend to produce larger seeds tend to produce smaller numbers of them, whereas those that produce smaller seeds tend to produce more of them. The trade-offs occur at all levels of biological organization. Just to be clear, definitionally, if we're on the same page, I use a relatively broad definition of trade-offs. Some people are much more narrow in terms of how they might define trade-offs. This is modified from Doug Fatuma's textbook. The advantage of a phenotype, and I say phenotype because it may be determined in many ways, in one ecological context, accompanied by some disadvantage in either this same ecological context or some other ecological context. We're often detecting trade-offs and studying them through the study of negative correlations between traits, between functional traits, traits that we expect or that we know to enhance fitness under some conditions. And I think that that's an important thing to recognize. Not all negative correlations are trade-offs because they may not result in advantages or disadvantages. But if the traits are in fact functional for the organism, then a shorthand to studying trade-offs can be finding a negative correlation between two traits. I think it's widely recognized that negative correlations and trade-offs can be driven by multiple factors. One of the things I'd like to argue today is that ecology, there's a balance between ecology and constraints of physiology or genetics, on the other hand, that may generate trade-offs in organisms. When I think of ecological drivers, I think of basically selection, natural selection operating, and the organism specializing in some way to cope with particular environmental challenges. On the other hand, there may be certain physical limitations or physiological limitations in allocation of a fixed amount of resources that can only occur one way or another, sort of that Y-tube where we think of constraints. I will make the argument that the negative correlations that occur at the species level or higher taxonomic levels are almost entirely driven by ecological drivers. That almost no constraint, of course there are some physical limitations, but very, very few, especially there are very, very few constraints that can't be broken at the level of macroevolution or across species. Perhaps counterintuitively, I'll make the argument that phenotypic plasticity, especially if it's adaptive plasticity, responses to the environment that maximize fitness, are a more intermediate balance between ecological drivers and constraints of physiology. Any individual organism, at any given moment especially, has some fixed pool of resources. When faced with an environmental challenge, the phenotype may change in some way to maximize fitness in that environment. So there's that level of ecological specialization. But because the amount of resources for that organism are essentially fixed for that moment in time, of course there's going to be some drain on that resource, and there are going to be constraints. I'll argue that genotypes within a species, genetic variation that we see, and negative correlations that we see within a species are most likely driven as alternative ways to allocate their limiting resources, driven by constraints of physiology and genetics. And I just realized of saying this, but Perhaps the exception is genetic polymorphism. So the example I gave earlier about the Micropterus and Macropterus insects, those, I assume, are different adaptive strategies that are maintained. But especially, I'd say, in cases where we don't see genetic polymorphism, but where we see negative correlations between traits, I would say that those are typically not adaptive, but really just alternate forms of allocation, weighing more heavily on the side of constraints of physiology or genetics. I hope more of this will become clear as we move through the talk. I want to emphasize that here's my sort of expanded Y-tube. It's a Y-tube of 1,000. By and large, I'm going to focus the talk on traits that are somewhat removed from or somewhat distant from direct correlates of fitness. So the big arrows for any organism are those fitness-related traits, or what are often referred to as life history traits, offspring size, number, growth strategies. And I'm going to try to focus my talk on the other little arrows, the 1,000 other things that all plants and animals do to interact with their environment in various ways. Ultimately, of course, they all contribute to fitness. Many of these little arrows contribute to fitness through offspring size, growth, other things. But they're the smaller traits that are a smaller drain on the energetic pool of the organism that ultimately cope with the ecology or the ecological challenges. All right, I'm going to take a quick drink here. Okay, so why are we interested in the evolutionary ecology of trade-offs? If I haven't convinced you so far, they help reveal constraints on organisms and their evolution. They link evolutionary history or the past to current ecology. Trade-offs yield diversity at all levels of organization. In other words, the genetic diversity we see within a species, or the phenotypic diversity we see within an individual genotype, so the species diversity we see they're all driven by trade-offs at some level. And they contribute to our ability to generalize and develop theory, given that all organisms face these decisions or trade-offs. The major questions in the evolutionary ecology of trade-offs that I want to address today are the following. I'm going to illustrate some of the ideas I have through my own work on plant-insect interactions in the milkweeds. And the questions I'll ask are the following. Do trade-offs associated with plasticity predictably shape community interactions? 2, what are the major intraspecific genetic trade-offs for expression of traits and why? And lastly, are the intra or within species trade-offs mirrored in inter or across species patterns? And if not, how are species strategies constrained? So we're going to kind of move across the hierarchy from plasticity to genetic variation within a species to across species strategies. So it's here that I get to introduce the natural history of the group I've been studying. And Linnaeus named the genus that the milkweeds are in Asclepius after the Greek god of medicine, the son of Apollo, in part because Linnaeus recognized both the toxic and medicinal value of the compounds that lay within the milkweeds. There is spectacular floral diversity. I'm not going to talk about the flowers, but I always like to show this image just because it's a relatively unique floral form. It shares with the orchids in that the pollen are packaged in packages called pollinia. One of the two major groups that have pollinia. You can see here it's a specialized floral form, but through natural selection has been stretched and recolored in various ways. In our region here in the Northeast, it's pretty easy to find 5 or 6 Asclepias species. There's 140 species in North America. And the sister group, Gomphocarpus, which people argue about whether it should be in Asclepius or not, this is a Gomphocarpus species. There are 200 species in Africa. The rest of these are all North American Asclepias. And they grow, they're typically open habitat species, so there's only a few species that grow in the shade, but they definitely prefer the drier habitats, but they do occur in mesic to the driest of the dry habitats. There are clonal forms, such as this species here from the Midwest, and there's spectacular diversity like there is in many, many plant groups. We've been able to study them both in the field as well as in common environments, so this is a living collection of about 45 species of Asclepias that we've had in the greenhouse. It's no longer in existence, but we resurrect it every once in a while. They're just a pain to try to keep alive and pest-free long-term in the greenhouse. You guys know all about living collections. The milkweeds are a great group for me in particular because I'm interested in plant-insect interactions, and there are a host of highly specialized herbivores that feed on different parts of the milkweed. These are the herbivores of the common milkweed, Asclepias syriaca, one that certainly probably grows outside of the building here. The diversity of herbivores is pretty neat. So here's a weevil, Curculiana, that feeds on the stems, the larvae inside the stem. There's a leaf beetle in the chrysomelidae, the larvae versus the adults that feed on the leaves. And then there's a cerambycid beetle, longhorn beetle, whose larvae, which do the most of the feeding, bore through the roots. So we're already looking at a stem feeder, a leaf feeder, and a root feeder. We have a leaf mining fly in the genus Liriomyza, so it's in the same genus as the greenhouse pest. 2 lepidopterans, a butterfly, the monarch, and an arctiid moth. This is the caterpillar. This is the same species that just hatched and when they're older. And then there's several species of sucking hemipteran insects, 2 species of seed bugs, and then there's 3 species of aphids, 2 of which are shown here. So on the one hand, a depauperate fauna, about 11 or 12 species, but taxonomically quite diverse in terms of the big orders of insects, and also quite diverse in terms of the gills, everything from boring through the roots and stems to sucking on the sap to eating the seeds to eating the leaves. Just as I'm getting a little off topic here, but you consider another common plant in the Northeast like goldenrod, and there's well over 100 species that complete at least part of their life cycle feeding on that plant, 40 of which are specialized. So milkweed and goldenrod grow in a similar environment, but this is a very depauperate fauna of highly specialized herbivores compared to something like goldenrod. Now I'm going to focus on 2 traits of the milkweeds as we think about trade-offs occurring through plasticity, genetic correlations, and species. And it's just really for brevity and so that I can get through things, I'm going to focus on these 2 traits. The first is latex. Latex occurs in about 10% of flowering plants and about 10% of both species and families, highly convergently evolved. The milkweeds, the dandelions, outside of the building here, they have latex. It's, of course, where we get our heroin from opium poppies. I knew that they were in Ipomoea, but I didn't realize that they're in the tubers of sweet potatoes, at least occasionally, lobelias, and of course, it's where we get our rubber. I think there are about 40, 45 plant families that have evolved independently latex production. Spectacular plant defense in terms of being physical, no known primary function for the plant in terms of acquiring or allocating resources. Its primary function seems to be defense. The other plant trait I'm going to focus on today are the cardenolides, or the cardiac glycosides, that Linnaeus knew about and that so famously make milkweeds toxic. Here's a generalized structure of the cardenolide. They're steroids. They have these 4 fused rings, and the carbon rings, and then they have a lactone up here on the top. And the way that cardenolides are poisonous is at once very general and also very specific. They bind to the animal sodium-potassium ATPase, or sodium pump as they sometimes call them. Every animal cell is pumping ions across the membrane using sodium-potassium pumps. And the cardenolide binds to those pumps and stops them from functioning. A very general toxin because any animal cell that comes into contact with cardenolides will be in trouble, but very specific because it targets only one very specific enzyme that animals have. I threw this in just as a brief digression just because it's such a cool story. Cardenolides are great because they're toxic and they make milkweeds poisonous to most herbivores, and the monarchs sequester them and all of that. In addition, they have a really remarkable story throughout human history, and I'll talk a little bit more about this tonight, but the leading hypothesis for why Vincent van Gogh painted the way he did, and in particular, why in the last 2 years of his life, Van Gogh's paintings took a turn for the yellow and the halo-y, is that his doctor, Dr. Gachet, which he painted twice holding foxglove plants that have cardiac glycosides, have cardenolides, he was probably prescribed foxglove tea, cardenolides, for his epilepsy. And what we now know is that those heart patients that take digitalis for congestive heart failure, when they get too much, they see yellow and they see halos around things. So the leading hypothesis is that 2 years before Van Gogh took his own life, he started painting, sunflowers and wheat fields and starry nights. That's when that turn occurred, was likely when he started taking digitalis for his ailments. But more on that later. Okay. To talk about trade-offs across biological levels of organization, to focus on cardenolides and latex, I first want to convince you of something that I think on average has to be true, but of course there are going to be exceptions. And what has to be true is that on average, trait variation should be hierarchical. That which is imposed by the environment through plasticity is going to be relatively low. If we grow common milkweed in different environments, the amount of latex that exudes might vary 20 to 40%. In other words, in shade or when attacked by herbivores, there's some plastic variation. If we grow the common milkweed in a common garden and allow different genotypes to express genetic variation, we see higher levels of variation, fourfold variation. And if we look across 45 species of Asclepias, all sharing a common ancestor in this group, there is even more substantial variation from those that we can't detect any latex, like the butterfly weed, the orange butterfly weed that some of you will know about, to some of the species from the Sonoran Desert which bleed the most. Cardenolides basically show the same pattern. Plastic variation relatively lower, genetic variation within a species greater, and species level variation even greater. We can think about generalizing that in some way if we have some trait axes, multivariate trait axes 1 and trait axes 2. We can think about these large circles as different species. They're phenotypically, on average, separated quite far from each other. We think of genotypes as these rectangles, and the space within those rectangles that can be explored within a genotype due to plasticity or environmental variation. Now, of course, I think it's sort of popular right now to think about trait variation being greater, let's say, within a species than across species. I don't really understand that. On average, it must be the case that variation across species is greater. Okay, so I don't know what time. I got to get moving here. Let's think about the trade-offs associated with plasticity. Here's the Y-tube. Plasticity, as we've already talked about, is the ability of a single genotype, when it passes through some environmental stimulus, to produce phenotype A versus environmental stimulus B, phenotype B. I'm mostly going to be considering adaptive plasticity, or traits that we think are being expressed in response to increasing fitness in those environments. And I want to focus on the inducible plant defenses that plants have, or trade-offs associated with induced plant resistance. That's a form of plasticity. I'm going to explain to you what that is. Here is the plasticity of the pattern of plasticity and resistance. I'm showing the amount of toxin that milkweeds make, common milkweed. When it's undamaged or healthy, relatively low amounts. When it's cut with the scissors, that amount doesn't change. When it's attacked by a monarch butterfly caterpillar, it may increase 20 or 25% 5 days later. There's a set of hormones that regulate that induction, and the nearly universal hormone that's been implicated in defense is jasmonic acid, or jasmonate. This is something that's now been studied very, very widely. The earliest plants, the earliest diverging lineage that we have, that we can study, basically produce a jasmonate burst following herbivory that is associated with the increased production of defenses. That might be increased amounts of latex and cardenolides, as in the milkweeds. It's increased extrafloral nectar in plants that have those. It's increases in glandular trichomes in plant species that have those. It really is, there's only 7 or 8 hormones in plants, or however many there are, and the main function of this one, jasmonic acid, is regulating defense. Of course, they do other things in collaboration with other hormones, but the main thing that jasmonate does is upregulate defense. The pattern of induced resistance is that when you grow a monarch caterpillar now on a previously undamaged plant versus a plant that had previous damage by a monarch, the caterpillars grow about 20% less. They're poisoned, by either the cardenolides or the increased latex or other traits in the plant, and they have a harder time. This pattern, again, is relatively universal. When people have studied plants that are damaged by herbivores or induced by jasmonic acid, herbivores have a harder time on those plants. Okay? Now, the world of herbivores on plants, however, is not singular. They don't exist only, it's not just monarchs and milkweeds, but there's the community that's out there. And there are often trade-offs associated with the expression of defense because different defenses are effective against different herbivores. In this case, we have a control plant or a plant damaged by a monarch. The amount of cardenolide increases. However, if there are aphids already feeding on those plants, as is shown here, monarch attack does not increase the cardenolides. I don't have time today to get into all the mechanisms, but I think we fairly well understand that aphids, again, this is not unique to milkweeds, but when they attack plants, they typically upregulate a different hormone, salicylic acid, which typically upregulates other defenses that are specific to phloem suckers, those that put their mouth parts and suck phloem. And the trade-off here is then that the plant can't simultaneously defend against both of these attackers. We can look at the ecological outcome. Monarch larval mass, here shown here on the y-axis, is higher when it's feeding on plants that have aphids on them compared to no aphids. Here the plant can't induce its jasmonate defense, doesn't increase its cardenolides and latex, and the caterpillar performs better. In this case, the trade-off is not symmetrical, or the interaction is not symmetrical. The plastic effect is asymmetrical. If we look at aphid population growth, it is reduced on monarch-damaged plants. If the trade-off was symmetrical, you might expect not only do caterpillars benefit from aphid attack, you might expect that aphids would benefit from caterpillar attack, but we see the opposite. It's a little bit complicated, and that's led the postdoc, Jared Ollie, who worked with me on this project. We wanted to title the paper, Aphids Suck and Monarchs Rule. But the editor of Functional, it was really clever, right? Aphids are suckers, and monarchs benefit from the aphids, but monarchs suppress the aphids. But the editor of the Journal of Functional Ecology didn't like the title, so he didn't want me to use it. The trade-offs occur in other contexts as well. Here's another community context of the herbivores. Here's the Tetraopes, the four-eyed milkweed beetle. When it attacks plants, compared to a control, this is the abundance of Tetraopes on the y-axis for this comparison here. It is attracted to damaged plants. And we now know that basically the Tetraopes beetle uses damaged plants as a mating source. They are attracted to those plants and then they find mates on those plants. So they're more attracted to damaged plants compared to undamaged plants. But a Tetraopes damaged plant signifies to a monarch butterfly this is not good food. We see reduced oviposition by monarchs on those plants compared to undamaged plants. So again, the interaction in this case, I don't know if I can't remember if it's symmetrical or asymmetrical, but there is a trade-off in terms of what is attractive to one herbivore compared to repellent to the other. I'm going to summarize the plasticity part of this. There's a lot I wasn't able to tell you. If we agree to consider adaptive plasticity, it evolves presumably due to variable environments. There's a diverse herbivore community in this case. It allows for the rapid management of phenotypes in the face of changing environments. The trade-offs may be predictable based on hormonal physiology and ecological drivers. I didn't get into the details of that. But again, as we do know things about the jasmonate versus salicylate signaling in the plants and who's attacking those plants. And basically, some predictability in terms of understanding who will be attracted or repelled when the plant induced defenses. And lastly, these plastic responses create predictable outcomes in community structure. I showed you sort of two pairwise things between caterpillars and aphids and adult ovipositing monarchs and Tetraopes. We have done other studies at more the community level, and when we set plants on particular trajectories, ultimately the community structure of insects on those plants is altered. Again, some are attracted and some are repelled. Okay, let's consider, we're back to the trait hierarchy here. We considered plasticity and the trade-offs that occur within a species or within even the genotype, within a genotype of a species. We now want to consider the genetic correlations or trade-offs associated among genotypes. Often when people are interested in studying the evolutionary ecology of trade-offs in organisms, we use common garden environments. In this particular common garden, there were 23 full sib genetic families of the common milkweed, randomized. There's about 20 individuals of each family randomized here. And so we basically are able to study the phenotypes of these organisms, of these plants. And the within family phenotypic variation can be ascribed to genetic variation because each family is represented at different parts of the garden. So the more similar they are would indicate the genetic component to those families. I first want to show you that, in fact, genetically determined traits are beneficial, are important to these plants. We talked about cardenolides in the last segment. I'll talk about latex here. The x-axis is the amount of latex expression. On the y-axis here is the oviposition by this highly damaging stem weevil, rhizomatus. Each dot in this graph represents a full sib genetic family of common milkweed with about 20 replicates in that same common garden. Now, the first thing I want you to recognize is that there is a tremendous amount of genetic or heritable variation. There are genetic families that produce 4 milligrams of latex when we break the leaves, and those that produce less than 2 milligrams of latex. And that's repeatable because we're measuring these 20 individuals that are spread throughout the garden. The other thing I want you to take away from this is that those genetic families of the common milkweed that have higher amounts of latex have reduced oviposition by the weevils. These weevils work very hard to get around the latex. They're often getting latex all over their snouts. If there's too much latex covering the hole that they drill, they will not lay eggs. They are deterred by high levels of latex. Okay, so what are the trade-offs associated with the genetic expression of latex? Well, one very popular idea through the history of plant-herbivore interactions and plant defense is that defenses themselves should trade off. If you have latex, it's redundant, presumably it's effective. We don't need to have the cardiac glycosides on the other hand. We simply don't find that negative relationship when we look across defenses. I'm showing you the example of latex and cardenolides. Meta-analyses across other studies have basically not found that redundancy theory predicted negative relationship between two defenses. The different colors and shapes here are just 3 different data sets. We have them in the field, we have them in the greenhouse, different kinds of collections. We never see a negative relationship between the amount of latex that's produced or the amount of cardiac glycoside that's produced. I should say that it's physically possible. We have milkweed species that have a lot of latex but no cardenolides and vice versa. So it's not simply the case that the cardenolides are all in the latex. It is possible, and we have certainly some low latex, low cardenolides, and we have some high ones. We have individual genotypes that basically do anything they want. Well, perhaps if we think about them as not negatively trading off, we might sort of ask the question, why? And if you think about the million, or at least 1,000 things that any organism does, defense is certainly one of them. These plants produce latex and cardenolides. They also make dense trichomes, which have to be shaved by some of the herbivores. And if we think about the diversity of organisms that feed on these plants, as we've already introduced at that community, not all of them are impacted by latex. The monarch butterfly is indeed impacted by latex. It often will spend 20 minutes or more cutting the laticifers, letting the leaf hang down before it then feeds on the plant. And in fact, all of the other chewing herbivores of milkweed deactivate or drain the latex as well. Here's Labiodomera. It cuts the distal veins and cuts the veins and then feeds on tissue distal to that is latex-free. That's what rhizomatus does. Here's a mating. The females typically cut, then they start eating here, and males fly in attracted to the damage. Here's an arctiid moth, cuts the veins before it eats. But there's an entire set of herbivores, as I mentioned to you earlier, that don't come into contact with latex, aren't negatively impacted by it, because they're sucking the phloem, they're eating the seeds, or they're mining the leaves, not coming into contact with laticifers. That community diversity dictates that one defense will not be effective against all the attackers. The same is true for the cardenolides. Milkweed insects have different relationships with the cardenolides. So here's an array of some of the more common milkweed insects, their genus names, and what I've put in to summarize just an incredible amount of work, whether we know them to be negatively impacted by the toxins, whether they have particular genetic substitutions that allow them to cope with the toxins, and whether they sequester them. And if we just go across the top, there are indeed some aphids and tetraopes, which we have evidence are negatively impacted by cardenolides, others not, and for the monarch, I'd say sometimes. That's our evidence. Several species have the same identical genetic substitutions, which make them relatively insensitive to cardenolides. Others do not. They deal with the toxins in other ways. Some of these species sequester those toxins for their own benefit, concentrate them in the body. Others do not. That community diversity allows the cardenolides in latex to function somewhat independently. So community diversity maintains variation for traits that might otherwise trade off if they were truly redundant. I didn't talk about this, but we have some evidence that defenses, in fact, may be complementary. Something I'll talk a little bit about tonight. In the life of a monarch butterfly caterpillar, it first shaves those trichomes. It then deals with those leuticifers. It tries to cut a little trench and make an island for itself to feed. And then it copes with the cardenolides in the leaf tissue that it's consuming. So especially when these organisms are young, they've just hatched, multiple defenses may be especially effective in preventing them from growing or completing development. Okay, so thinking more about intraspecific trade-offs, where do they occur? If we go with the notion that they are mechanistically dictated by physiology or plumbing or pleiotropy, ultimately traits that are connected in that way may trade off. Ecologically, if traits are ever truly redundant, we may expect trade-offs. Or in the case where ecologically, when a genotype experiences one environment and is simply not exposed to some other challenge, some ecological driver, they may negatively trade off. What I'd like to consider in this part of the talk as a third explanation is that when there's a fixed pool within a species of some resource, genotypes may simply represent different ways to allocate that resource. So we're thinking about genetic trade-offs, but I want to return to the plastic strategy of inducible defense or increased cardenolides when a plant is damaged compared to when it's not damaged. And I'm going to focus on this parameter we'll call inducibility. It's the amount of increased toxin in the damaged state compared to the control state. A long-held prediction in plant defense theory has been that different genotypes that allocate more to constitutive defense or defense up front in the absence of damage will have reduced levels of inducibility. If you make a lot of defense without damage, after damage, you should not increase very much. Whereas those genotypes that produce very little constitutively without damage, once damaged, they should increase a lot. This is based on the notion that that within a species there's a fixed amount of cardenolides that's produced through metabolism, and genotypes may alternatively allocate that up front or after damage. And indeed, when we've looked across, in this case, 20 different genetic full sib families of the common milkweed, those genotypes, each dot here represents a genetic family with multiple replicates. Those that produce a lot of cardenolide up front, not only do they not induce a lot, they actually reduce the cardenolides in the week subsequent to damage, whereas those genotypes that produce very little upfront after damage increase substantially. So we find evidence of this negative genetic correlation between constitutive and induced allocation. We find that also in the roots. This here is not drawn to scale, but this is the larva of the four-eyed beetle. It's a specialist on the roots of common milkweed. And those genotypes that make a lot of cardenolide in the roots after damage again decline, whereas those genotypes that make relatively less upfront after damage by the root beetle, they increase the amount of toxin that they make. So for trade-offs among genotypes, they may be determined by allocation within that species strategy. Common milkweed on average makes around 1 milligram per gram dry mass cardenolide, and different genotypes allocate that constitutively or induced. We can ask the question whether those same trade-offs then can scale up and occur across species. And here I want to introduce introduced the null hypothesis, which was produced by, it was, I'd say, formalized by Dolph Schluter in a, just I think, what a really forward-looking paper in 1996, adaptive radiation along the genetic lines of least resistance. And here Schluter made the argument is that if genetic correlations which occur within species, as I've shown you for common milkweed, a trade-off between constitutive and induced resistance, if basically macroevolution occurs along that same line, then the null hypothesis is that species that make a ton of cardinalides that evolve basically will slide up and down along that same genetic correlation axis. Sort of the null hypothesis of how evolution should occur at that larger scale. What we did is we took 50 species of Asclepias. We had obviously less replication within each of these, but several individuals. Each dot is a milkweed species. We plot constitutive cardinalides on the x and inducibility on the y. You'll remember that our prediction and finding from the common milkweed was a negative relationship, but this relationship is positive. That threw us for a little bit of a loop. We looked at the root beetles, attacking, in this case, 18 species of Asclepias, and for root cardinalides, we see the same pattern. High constitutive cardinalides, after damage, a greater increase. That counter-gradient pattern held across species for both leaves and roots. Okay, well, this led us to try to reconcile the within versus between species patterns that we saw. And it led us back to a classic paper by Van Noordwijk and de Jong from '86. It's something we kind of conceptualized for induced defense in the book chapter, where we made the prediction that for common milkweed, we saw this negative genetic correlation between constitutive and induced within, but across species, we saw this pattern. And it led us to the notion that basically macroevolution may occur perpendicular, where as reproductive isolation occurs, species are not necessarily sliding along this axis, but are changing so many things about their overall metabolism. As macroevolution occurs, the size, the amount of leaf area, the root-to-shoot ratio, the habitats that these organisms are living in can change. And so we predicted that within any given species, we may see a negative genetic correlation that represents alternate forms of allocation, but that in this case, across species, we see the positive correlation. We've basically now done this for 6 or 7 species. Here's Asclepias fascicularis from California, 20-odd genetic families within it. It's an extremely low cardinalide species. It shows that negative correlation between constitutive and induced across families. We can take Asclepias linearia from the Sonoran Desert, again, 20-odd genetic families, not as steep a slope, but again, a negative genetic correlation between constitutive and induced. And now, if we fill this in, I'm not going to show you all the negative correlations, but they're basically all negative within a species. So our summary here is that within a species, there is a defense trait ceiling. There's a strategy for that species. And the trade-off occurs among genotypes in terms of how they allocate those cardinalides, up front or only after damage. Genotype dictates the level of plasticity within that species. Across species, macroevolution does not occur consistent with Schluter's null hypothesis along the genetic lines of least resistance, but in this case, overall cardinalide metabolism evolves. And where there's greater overall metabolism, inducibility proportionally increases. Okay, I got 5 more minutes. Sorry, guys. Let's consider what trade-offs do occur then at the species level. And here I want to introduce the notion that strategies evolve due to ecological specialization at that macro scale. Plant species that live in particular habitats or that are attacked by particular guilds or communities of herbivores will convergently evolve and defend with repeated sets of traits. This is, for those of you that know plant ecology, if you think about Grimes' triangle, what was Grimes' triangle? It was ecological specialization to particular kinds of strategies. There are trade-offs that occur at that level. Competitors don't do well under stress in Grimes' triangle. We're making the same argument here. I show you this picture of giraffes because I haven't considered really vertebrate herbivores as being important for Asclepias. but we expect that if the major herbivores of species are vertebrates, or that the habitats are radically different, that the plant species will specialize, and the trade-offs we'll see will occur at that macro scale. And this builds on the classic work of Paul Feeney, Lissy Coley, and Judith Becerra, who basically considered these ideas in that macro scale. I'll just say that I was inspired to think about plant defense strategies or syndromes when I sat on a dissertation committee at the University of Toronto. There was a graduate student there studying pollination syndromes, and in the genus Penston, and I just remember sitting there just being blown away. 14 or 20 times independently, the plants have evolved hummingbird pollination from bumblebee pollination. The traits basically are a set of correlated traits that go together. It's the blue flower with concentrated nectar and the anthers in there, the dilute nectar of the red flower that hangs down. And I sort of thought, well, okay, if there are ecological conditions that drive this kind of thing, we visually see it as a naturalist. But as somebody that studies defense, we don't always see the traits that might be functionally defensively and that might evolve together to form syndromes. So in this case, we looked at 25 Asclepias species in a phylogenetic context, measured 7 traits that were at least contribute in some ways to defense. What we found was that of the possible 21 pairwise correlations, we found no negative correlations across those species, but 5 significant correlations, and they were all positive. Traits are covariant, resistance traits are covariant positively. This was convergent. Again, we have these clusters in multivariate space where species repeatedly evolve into clusters of defense space. This fits some with the kind of defense strategy classic literature that Paul Feeney, Coley, and others developed, where we, in this case, have a x-axis that's barriers to feeding or toxicity, and digestibility or edibility on the y-axis. I won't get into all the details here, but basically milkweed species clustered into those that seem to escape or tolerate, those that provide no low nutrition to herbivores, and those that have a balanced nutrition and defense. We didn't find species that were up here or down here. Some of the positive correlations are really interesting. So for example, across species of Asclepias, the amount of latex that is exuded and the trichome density is positively correlated. We have no reason to think that there might be physiologically linked hairiness and latexiness. Within a species, we don't find genetic correlations between latex and trichomes. And even here, the exceptions are really interesting. They help prove the rule. We now have 5 species that are very high in latex, but have no trichomes. It turns out that these are the only 5 species that have thick layers of epicuticular waxes. And what we've now understood is there's something about covering the leaves. And the waxy species are physiologically, in terms of photosynthesis, very similar to the high trichome species. And basically, high latex, you're either really hairy or you're really waxy. But if you're waxy, you have no hairs. And so there's just some really interesting biology in there that is yet to be figured out. Resistance in this case, the trade-offs occur at the macro scale of strategy. In this case, resistance and tolerance, the ability of a plant to regrow following herbivory trades off with the toxicity. So here's the ability to regrow on the x-axis, toxicity of the y-axis, each dot is a species, and these two show a weak but negative relationship. I'm going to summarize across these levels of organization now. I hope I've convinced you today that trade-offs are indeed ubiquitous, they're important, but they're not the same across different levels of biological organization. I'd say as a methodological note, we ought to be very careful if trade-offs are expected due to plasticity or some hormonal physiology. At that level, we shouldn't necessarily expect it to occur at the genetic or species level. Mechanisms and ecology help us to solidify our predictions about trade-offs. If we understand something about what's limiting or how hormones work, or the ecology, what is experienced by one genotype or species versus another. Plasticity comes with trade-offs but allows flexibility and is determined by the genetic starting point. Genotypes within a species are constrained by the species' strategy overall, and often will represent alternative allocation phenotypes. And species trade-offs occur at the level of multivariate strategies, and most all bivariate trade-offs, I would argue, are in fact breakable if selection is strong enough. Lots of people to thank. I'll just did a lot of this work. I'll leave this picture up here for a second, and thank you very much for your attention. Happy to take questions, and I'll leave these take-home messages up. Go ahead.
You're offering trade-off between constitutive and inducible defenses. It would always be an advantage to do both, right? Unless there's an energetic or epigenetic constraint to that.
So the question is why? Is it why?
Well, I mean, would you say macroevolution goes towards maximizing both things?
Yeah, I guess what I'd say, and
I was just surprised that there was
yeah,
a trade-off.
it's interesting. I think two things about it. One is there very well may be, like metabolically, if a common milkweed makes a milligram per gram dry mass cardenolide, I guess to make twice as much of that may be impossible. But the other thing that I've thought some about, and I've thought a lot about this because the first time we saw those negative ones where, genotypes that have high constitutive, after damage they decline in cardenolides, that seems to be fairly repeatable. We always have genotypes that are high, but after damage go down. I guess if I was to put a human brain inside of a plant, like maybe the plant is thinking, I've put up all of these cardenolides and the monarch is still feeding away. Clearly that's not effective. Not only am I not going to produce more, but I'm going to do something else. Now, that's a lot of anthropomorphism, but I suppose if the metabolic machinery is telling me only to make a fixed amount that's this much anyways, and I'm getting fed upon, I suppose do something else is not a bad thing to do.
It's also possible that in that same situation where the plant gets attacked and is munched and then it drops down this cardenolide, it's making something that we don't know.
Definitely.
Totally beyond what has been looked at yet.
Definitely. I mean,
and maintaining the same energetic approach to trying to disrupt the predator. But
these plants, and I'm not talking about like milkweeds. When I say these plants, like all plants, I mean lettuce. They're just making tons of different compounds. I love latex and cardenolides, but these plants are making cysteine proteases. They're making saponins, and they're making a bunch of stuff that I haven't considered. Definitely.
So when you find that you don't see a trade-off like between the latex and something else, do you think that that's because the trade-off is with something you're not measuring, or do you think that there really is no trade-off in defense there?
Yeah.
That's what I'm getting at is there's often this idea that you should be locally adapted to do this or adapted to do this, but why not just be good at both if you can?
Yeah. I definitely think that it's the case that, I guess I believe in the universality of trade-offs.
Okay.
If there are 1,000 arrows, which I think there are in terms of what organisms allocate to, it seems that there must be some other arrow that declines with increased allocation to some other arrow, because I think there are these energetic, limitations. But I think of herbivores. Okay, there's pathogens, there's the competitors, there's repairing yourself, there's preparing for reproduction. And that's just like the big stuff that we think about. There's the microbial interaction. So I'm not sure how useful it is. It's a little bit like the competition theory from the 70s, like when we didn't find competition, it's like, well, we just didn't look hard enough or look at the right competitor. My guess is you can find the thing that trades off, and the really interesting questions are why, and is it predictable? Yeah. Not sure if that answered the question, but.
Is there any correlation across species so are plants that are good to resist sucking insect bad to resist chewing insects, or there is no correlation?
That's a good question. I don't know if that analysis has been done. I mean, it's certainly not been done for milkweeds. The yellow aphid, Aphis nerei, is on 6 continents, and it eats basically, not all Apocynaceae, but certainly eats all Asclepias. And most of them have chewing herbivores also. There's some evidence that resistance to pathogens and herbivores trades off across species, but I'd say that it's, and genotypes, but the evidence is most strong as a plastic response, where when plants upregulate jasmonate, they tend to downregulate salicylate. Yeah.
Any other questions?
Anybody know the genus of the plants on my shirt? Very proud of this shirt. There's 4 more here. They grow in the same habitats. They're pachypodiums. They're Asclepiae. They're Apocynaceae. Anyways, thanks a lot. Perfect.